Thanks for joining today. I'm Dr. Helen Huang, and I'll be talking about understanding the good, bad, and ugly of using electroencephalography, EEG, and new frontiers for EEG. I'm an assistant professor in the Mechanical and Aerospace Engineering Department, and I'm part of the UCF Bionics Cluster and the Disability Aging and Technology Cluster. Some of my key achievements here at UCF include an NSF Career Award, an NIH R01. I'm an alumna of the National Academy of Engineering Frontiers of Engineering program. And more recently, my student and I won the American Society of Biomechanics President's Award at the 2020 virtual meeting. And one of my students' papers was selected as a Frontiers in Neuroscience Brain Imaging Methods Editor's Pick. My research focuses on neuromechanics, the interplay of neuroscience and biomechanics. We're interested in human locomotion, so walking and balance control. We're also interested in locomotor adaptation, so the process of adapting walking patterns. We perform mobile brain and body imaging experiments, and we have an interest in gait rehabilitation and healthy aging. I've taught several biomedical engineering courses here at UCF, including the undergrad and graduate biomechanics courses, bioinstrumentation, and topics in biomedical engineering. So I direct the UCF Biomechanics Rehabilitation and Interdisciplinary Neuroscience Lab, or the Brain Lab, and our mission is to advance knowledge about the human brain and human locomotion, and to develop new methods for making brain technologies more accessible and inclusive. And to advance knowledge, I believe we need to critically challenge methods and perspectives in our research fields. And to this end, I've identified what I consider to be the good, bad, and ugly about EEG as of now. So EEG is the recording of electrical signals on the scalp. And my approach uses methods and algorithms that were developed by others and used by many to estimate the sources of that electrical activity in the brain, and then to analyze the characteristics of those brainwaves. And the first results of EEG source localization of gait were published by Gwynn et al. In 2011. And other studies on healthy young adults over the decade have identified similar areas, namely the left, right, central sensory motor cortices, parietal cortex, and the anterior cingulate as having a significant role during walking. And in my lab, we have also shown that these areas are involved when exercising on a recumbent stepper while responding to perturbations, which suggests that locomotor tasks like recumbent stepping could be used to engage cortical processes also involved in walking. So I think the consistent EEG sources found in gait studies is a good thing about EEG. But EEG is known to have poor spatial resolution. And the algorithms for identifying these cortical sources can be influenced by other factors. For the paper that was selected as one of the editors picked, we had evaluated the reliability of different methods for digitizing the locations of the EEG electrodes. And we found that less reliable methods like ultrasound that has more variability resulted in having more source uncertainty. Interestingly, we also found that with our general broad spatial targets, any digitizing method we tested generally resulted in sources in the same Brodmann areas. So a new frontier for EEG would be to find ways to not just improve our confidence in the sources identified, but to improve spatial resolution to be more specific, such as being able to confidently distinguish between the leg and the hand areas in the sensory motor cortex. Now, something bad about EEG is always wondering, is it really the brain or is it something else? And this is actually an age-old problem ever since the first description of EEG in humans by Dr. Hansberger in the early 1900s. And the problem is that EEG electrodes record a mixture of electrical signals. So you have signals from the brain, but also from eye movements, muscle, and the heart, as well as electrical signals outside of the body, such as power line interference and electromagnetic interference signals, as well as movement artifact. And during walking, movement artifact signals are a major challenge. And in a previous study, my colleagues and I, we did what we call the WIG study, which was to analyze isolated movement artifact signals. So to do this, what we did is we used a swim cap to block the electrical signals from the body, such that the electrical signals we recorded would be primarily the movement artifact signals. Then we processed this WIG data as if it were traditional scalp EEG data to see where the sources would be found. And while there are not that many sources in the brain volume, there are sources in problematic areas that overlap with areas that are found during walking. And if we look at the time frequency characteristics, there's some resemblance between what is only from the WIG and then also what is reported as being the brain. So this has led to a new frontier, dual layer EEG, where the traditional scalp EEG is recorded simultaneously with isolated movement artifact signals using a dual-sided electrode and a conductive cap. So in my lab, we've developed a 128 electrode dual-sided electrode system. But because it's such a new approach, there's many questions about what is the relationship between the signals of the two sides of the dual-sided electrode. And so as part of my career award, we're performing benchtop tests to characterize the relationship between the signals on the dual-sided electrodes. And with this knowledge, I believe we can improve the unmixing of EEG signals into their sources, and then leverage knowledge about the sources to estimate other measures that would be of interest for neuromechanics, such as gait symmetry, eye gaze fixation, and metabolic energy costs. So in addition to the brain dynamics. And this way, EEG alone could be used for multimodal neuromechanics. All right, now the ugly. The inadvertent exclusion of Black folks and other folks with curly, tightly coiled hair. While EEG has been an immensely helpful technique to understand brain function, imagine not having access to medical or clinical treatments and technologies that are based on EEG because of your hair. Or imagine being asked to shave your hair for an EEG treatment. So as you've probably come to realize, recording good EEG signals requires having good contact with the scalp. And folks with curly and tightly coiled hair often pose a challenge for recording good EEG data. And this can lead to longer prep times for those folks, poor EEG signal quality that results in their EEG data being excluded in the final analyses. And in some cases, folks with curly, tightly coiled hair may not be recruited or tested at all. So because curly and tightly coiled hair are often found more commonly on folks from the Black community, Black folks are disproportionately excluded in EEG findings. And this is a topic that has gained some attention recently. During Neuro-Racism Day of Black and Neuro Week, they had a discussion about EEG research and hair. And included on this panel is one of my own students, Liesl Richardson. She's a biomedical science Ph.D. student. And she's also one of the co-founders of Black and Neuro, which was an immensely, hugely big success. And we're also collaborating with Nina Woodley, who is a curl expert and Black hairstylist in Orlando to help us with our project. In addition, a recent theoretical perspective paper on systemic racism and EEG research was published and highlighted on Twitter that got some attention, and Liesl's tweet about what our efforts are in my lab to address this problem received many likes and retweets as well. So this is the last frontier for EEG. I want to mention the EEG, the idea of EEG and neurotech for all. So while some groups are working on developing new hardware, such as new electrodes to work with curly and tightly coiled hair, such as the Sevo electrode from Carnegie Mellon, my lab's approach is to focus on developing guidelines for EEG hair preparation to provide options for participants and researchers for working with diverse hair types, instead of working on developing new hardware for people to adopt. So we're working with, again, a local Black hairstylist and curl expert, Nina Woodley, to help us develop these guidelines. And we're also working with Dr. Jennifer Sandoval and Dr. Lindsay Neuberger in the Nicholson School of Communication for help with communicating with underrepresented communities and doing survey research. And at this stage of the research, we're trying to gather information from the EEG community. both researchers and participants, to help us develop comprehensive guidelines, as we are not the all-knowing experts on working with diverse hair types in EEG. We have one system, we have a limited number of Black folks to help us develop these. So we need the whole community. I invite you to please visit the project webpage at hellobrainlab.com/research/EEG hair project, or you can use the QR code on the screen to view the guidelines and be part of the solution and to help do the survey. So based on the good, bad, and ugly of EEG, I've identified 3 new frontiers for EEG. 1, improve spatial resolution and confidence in sources. 2, dual layer EEG and leveraging information in the EEG artifacts for multimodal neuromechanics. And 3, making EEG accessible to and inclusive for all. And again, it will take the whole EEG community to pull our tips and our lessons learned, et cetera, and love to get more people to do the survey. And we will compile the knowledge shared, and we'll update the guidelines for EEG and hair preparation as a resource for the entire community. Thanks for listening.